on the oxidative phosphorylation reactions and the electron transport, we have understood how each complexes of the electron transport act and how they are passing the electrons to the final molecule electron acceptor, that is oxygen. And in between, when these electrons are getting transported, you've also seen how the proton pump or the proton gradient is established and how ATP has been generated in the video. If you want to, please refer to it once again before we start this next topic, because if that concept is not clear, this balance sheet of ATP is going to be a little difficult to understand. Now, for those who have understood or got the concept of oxidative phosphorylation, electron transport and chemiosmotic hypothesis with respect to respiration, let us continue. So in this balance sheet of ATP production, we are going to see how those 36 ATP that one glucose molecule produces in aerobic respiration is coming from, and where the ATPs are coming from. So let us start with the first one, that is glycolysis. Now in glycolysis, as I've mentioned, there are two substrate level phosphorylation reactions. Okay, what is it? It is the 7th step. This one is the 7th step, and there is also the 10th step. In the 7th step, 1,3-bisphosphoglyceric acid is converted to 2-phosphoglyceric acid, where it is producing 2 ATP, and phosphoenol to pyruvic acid conversion is also producing 2 ATP. So substrate level phosphorylation of glycolysis is producing a total of 4 ATP. But we also know that in glycolysis, ATP is consumed. In the first step, which will be the first step of glycolysis, that is conversion of glucose to glucose 6-phosphate, and the third step, which is called as the isomerization, that is fructose 6-phosphate, another phosphorylation. When fructose 6-phosphate is converted to fructose 1,6-biphosphate, you have 2 ATPs that are used. Okay, so when we combine that with the 4 that we have gained through SLP, and when we use this, the net gain of ATP is only 2 in glycolysis. So glycolysis has only 2 ATP that has been produced. Okay, similarly in glycolysis there is NADH that is generated, right? When G3P is getting converted into bisphosphoglyceric acid, NADH molecule is produced, and we have also discussed this in the electron transport topic, that this is called as cytosolic NADH because this is coming from the cytoplasm, and we know that each cytosolic NADH is worth 2 ATP. I've already discussed how you are getting this 2 NATP. If you are still having doubts, go back to the previous to that video of electron transport systems, and you can come back and answer, and you can continue with this. So each cytosolic NADH is worth 2 ATP, and as a result, if you are using 2 ATP, NADH, if you are getting 2 NADH in glycolysis, then it will be 2 into 2, 4 ATP. So entire glycolysis is producing 6 ATP. 6 ATP means what? 4 from NADH and 2 from SLP. So glycolysis is worth 6 ATP. We'll consider it as equation A. The next one is the linked reaction, that is oxidative decarboxylation of pyruvic acid. Now in this, when pyruvic acid is converted into acetyl-CoA, you are forming 2 NADH, and this NADH is mitochondrial NADH because this reaction will be happening in the mitochondria. And I've already discussed that each mitochondrial NADH is worth 3 ATP. Again, in the electron transport system, in the electron transportation and the chemiosmotic hypothesis videos, we have discussed this in detail. If you are not able to recollect, please go back, revise and come back again. So each mitochondrial NADH is worth 3 ATP. So we are forming 2 NADH. So that will be 2 into 3, 6 ATP, and we will take this as equation B. Now, next will be Krebs cycle or the citric acid cycle. Now, citric acid cycle, there is one substrate level phosphorylation equation that happens where succinyl-CoA, when it is getting into succinic acid, you are producing 2 ATP. This is direct. Okay. Other than this, there will be NADH also that have formed at 3 different levels. What are those 3 different levels? They are our oxidation 1, oxidation 2 and oxidation 4. So oxidation 1, oxidation 2 and oxidation 4, all of them produce 2 NADH per 2 acetyl-CoA. And these NADH are mitochondrial, and we already know by this time, and I hope you're all perfect with the idea of how each mitochondrial NADH is worth 3 ATP, because each mitochondrial NADH is worth 3 ATP. So you are getting 2 NADH in oxidation 1, that is worth 6 ATP. 2 NADH in oxidation 2, which will again give 6 ATP, and 2 NADH in 4th oxidation, which will give 6 ATP. So you are getting a total of 6 plus 6 plus 6, 18 ATP from NADH. Next is FADH. There is only one step where succinic acid, when it is becoming into fumaric acid, it will produce FADH. And we know that 1 FADH is worth 2 ATP. So here you are getting 2 FADH. So 2 FADH is again worth 4 ATP. So when I take the ATP value of Krebs cycle, that is from SLP, from NADH and from FADH2, then it is 24 ATP. How 24? 2 from SLP, 18 from NADH and 4 from FADH2. You are getting 24 ATP and we will take it as C. So when I combine A plus B plus C, then it is 36 ATP, because A, that is coming from glycolysis. Glycolysis is giving 6 ATP. B is the linked reaction. The linked reaction is giving how many ATP? Again 6 ATP. And C is Krebs cycle, and Krebs cycle is giving 24 ATP. So 6 plus 24 is 36 ATP. Okay, in some books they don't consider the net gain of glycolysis. Then that means it will be giving 38. And one more thing, this is for plants, because in plants there is that separate as I've already discussed in that particular video of chemiosmotic theory, that plants have a mitochondrial NADH dehydrogenase which moves freely in the matrix, which directly breaks down the cytosolic dehydrogenase, and that's why there you are getting 2 NADH. But in animals there is no such free moving NADH dehydrogenase. So the NADH from glycolysis of the animal cell has to come into complex 1 just like any other mitochondrial NADH. So there again, instead of 2 you will get 3. So for animal cell, then this equation will change to 38 ATP. This is for plants. But in general, we generally study 36, because even in you are going to consider the net gain and everything. So this is the ATP balance sheet. Now let us see some questions that are associated with it. How many ATPs are generated in respiration? 36, 38, 12 and 4. Guess the answer? I don't think you need an explanation for this. It's a very direct question. I hope you all guessed the answer. So here is the right answer. Okay. The second question is, each mitochondrial NADH releases how many number of ATP molecules? 3, 2, 4 and 5. This is also another direct question. We have been discussing it quite some time throughout this video and the previous videos also. So each mitochondrial NADH is worth 3 ATP. Next question. ATP synthesis in mitochondria is closely associated with what? Release of carbon dioxide, consumption of oxygen, reduction of NAD and FAD, and release of oxygen. So what is happening here? Mainly it is reduction of NAD plus FAD, but those electrons are finally going to the oxygen. So the final answer is consumption of O2, because when these electrons go to the oxygen, only during that time you are able to establish the proton gradient. If it is just a reduction, then it is not going to create any problem, any point. And another thing, this is NADP plus, not NADH. Please remember, NADP comes only in photosynthesis. In respiration, it should be NADH. So internally also the question has this little problem. And the second one is reduction should be for FADH2, not for FAD. Okay, so that there will be that little confusion if you are not very clear with the topic, but this is the clarity. Next, net gain of high ATP in glycolysis per glucose molecule is represented as, well, here is the question. So what is the correct answer? It should be first. Why? Well, technically speaking, SLPs give you 4 ATP. NADH gives you 4 ATP. So you get 8 ATP. If I remove the 2 ATP that are consumed, then it should be 6 ATP, which is the net gain. And this one is the gross. So net gain will be 8 minus 2, that is 6 ATP. So let's see the next question. The transfer of 2 electrons from cytosolic NADH directly to ubiquinone pool is transmitted by. So this is something that has come from our question at the chemiosmotic hypothesis. So who is facilitating it? NADH ubiquinone reductase, succinate ubiquinone oxidoreductase, external NADH dehydrogenase, ATP synthetase. I have just mentioned this. Mitochondrial matrix will have 2 NADH dehydrogenase, specifically plant mitochondrial matrix. It will have a freely moving external NADH dehydrogenase. What it does is, it makes sure that the NADH that is coming from cytoplasm, the cytosolic NADH, it does not go to complex 1. It directly breaks it down and the electrons is directly sent to the ubiquinone. So the correct answer is external NADH dehydrogenase. Next question. Total number of protons contributed to proton concentration gradient is by mitochondrial NADH, cytosolic NADH, FADH during complete oxidation of 1-glucose. So this is a question that you can answer there or here also. That is, there means in the electron transport. So let us answer. Now I've told you that mitochondrial NADH. Well, mitochondrial NADH, each mitochondrial NADH gives 10 protons. Okay, 4 plus 4 plus 2. So there are a total of 8 mitochondrial NADH. So that means their contribution is 8 into 10, 80 protons. Next, cytosolic NADH. Cytosolic NADH, one cytosolic NADH is worth only 6 protons. 2 plus 4. So how many you have? You have 2 NADH. So it will be 2 into 6, which is 12. Next, for the third one, FADH2. One FADH2 is also associated with 6 protons. So here you are having 2 FADH2. That means it is 2 into 6, 12 protons. If you are not able to recollect how we have got this, I sincerely request you to again go back to the chemiosmotic hypothesis and the electron transport, oxidative phosphorylation electron transport mechanism, where how these protons have come, what is the mechanism, how many they have come, all this we have discussed much in quite detail. It will give you a complete picture. So the correct answer is 80, 12, 12. So option number 1. So the next question, number of protons. Okay, we'll just continue it in the next video because time is up for this one.
For more content, like, share and subscribe to www.i-win.in.